not physically there and only virtually here. But yeah, I hope your questions will come through the talk. Okay, so I'll start. So as I was introduced, my name is Noa Moriel, and I'm a PhD student at the Hebrew University of Jerusalem. And today I'll be speaking to you about my work with Dr. Edwin Mehmet and my PI, Dr. Mor Nitzan, on optimizing the allocation of resources for inferring the trajectory of cells. Okay, so let's start with some background. So cells undergo temporal processes such as embryonic development, where cells differentiate and acquire specialized functions. Single-cell RNA sequencing is a powerful tool that captures gene expression profiles of individual cells at a specific moment. Since cell populations are heterogeneous, for dynamical processes, changes in gene expression can reflect real-time developmental progression. Based on this assumption, trajectory inference methods attempt to assign a pseudotime to each cell as an indicator of its progression along the process. Many of such trajectory inference methods often start with standard preprocess, followed by calculating gene expression distances between cells, specifically shortest path distances in a reduced representation. Our focus here is on designing single-cell RNA sequencing experiments that optimally reconstruct such cellular trajectories. Can we determine the experimental parameters that preserve such cell-cell distances, presumably also leading to accurate depiction of trajectories? Okay, so more concretely, single-cell RNA sequencing experiments are typically constrained by a sequencing budget due to financial or technical limitations. That means we have a fixed sequencing budget, B, that needs to be equal to the number of cells we assay, and C times the average read coverage per cell, NR. As an experimentalist, given such a fixed budget, how do we determine the optimal number of cells to assay? Should we deeply sequence a few cells, like in one, or acquire a broader and shallower sample of cells, as shown in 3? Okay, so our choice of breadth-depth allocation can significantly affect the accuracy of the inferred cell-cell distances in gene expression space. We use geodesic or shortest path distance over the cell's KNN graph as the distance between cell pairs. The normalized cell-cell distances of the complete dataset are considered as the ground truth and are compared to the normalized cell-cell distances of some subsample data, which we refer to as the inferred distances. We define the reconstruction error, marked as epsilon throughout, as the average difference between these distances. Our objective is to identify the optimal breadth-depth allocation, that is, the number of cells and the corresponding average number of reads per cell, that minimizes this reconstruction error for a given sequencing budget. Our approach involves first modeling the individual effects of subsampling cells and subsampling reads, and then using this to explain the empirical reconstruction error observed along the breadth-depth trade-off for a given sequencing budget. This allows us to determine how the optimal number of cells varies with the sequencing budget. Okay, so let's start with an example dataset. So here we observe the differentiation of mouse embryonic stem cells into primitive endoderm cells in gene expression space, and the colors indicate time points in which the cells were assayed since induction. First, we model the read reconstruction error, epsilon t. We vary pt, the fraction of sampled read counts, and compute the read reconstruction error over 10 repeats. We plot both the mean and the variance of the error in gray. And overall, we observe that the reconstruction error decreases linearly with log PT. we consider the range of large PT to be a regime of saturated sequencing and model its read reconstruction error as constant. So you can see the fit of the read reconstruction error plotted in the dashed black line. Similarly, we vary the fraction of sampled cells, PC, and compute the cell reconstruction error, epsilon c. We observe that the reconstruction error decreases logarithmically with the number of cells. These two trends suggest that with a fixed sequencing budget, the reconstruction error can be non-monotonic across the breadth-depth trade-off. So that is, we can get high reconstruction error when both sequencing very deep or very broad. To explore the trade-off between breadth and depth, we compute the reconstruction error across a range of sequencing budgets. So near the y-axis, we have samples with few cells and many reads, while towards PC equals 1, we have samples with many cells but few reads. At low sequencing budgets, the reconstruction error is non-monotonic as a function of PC and is minimized at small sampled cell fractions. So for example, in pink, we show the breadth-depth trade-off for a budget of 60K reads, where the reconstruction error is minimized when sampling 60 cells with 1,000 reads per cell on average. As the budget increases, the optimal number of cells to assay increases. The empirical optimal fractions of cells that achieve the minimal reconstruction error are marked by colored stars according to their sequencing budget. By approximating the reconstruction error using the dominant error of the linear fits of the individual read and cell reconstruction errors that we saw before, we find that there is a parallel relationship between the optimal cell sampling probability and the sequencing budget. Our approximations are marked here in black stars for each project. So both estimates demonstrate that the optimal number of cells increases with the sequencing budget following a parallel relationship. Okay, so till now we just focused on a single data set, but our results actually repeat over multiple data sets that we present in the paper. And the data sets, they vary substantially in their biological features, including their differentiation context and the topological structure of the trajectory, as well as in their technical features, including the sequencing method, the number of acquired cells, and the number of reported genes. Our results consistently show robust trends across these datasets, validating the general applicability and reliability of our results. Okay, so last in our paper, we examined the breadth-depth trade-off effect on downstream task of recovering the temporal gene expression pattern of highly expressed and variable genes. So to do that, we decomposed this task into two sub-steps of first recovering the correct order of the cells and then preserving the gene expression signal in the data. So for example, for the mouse embryonic stem cell dataset at a budget of 60K reads, we show here the same reconstruction error curve as before, while highlighting sampling experiments of minimal reconstruction error in green and of deeper or broader choices in red or in blue, respectively. We use a diffusion-based algorithm to order the cells by pseudotime. We order both the full data and the subsample data and measure their ordering agreement in Pearson correlation. With Pearson correlation, we find that using the sequencing budget to sequence many cells at low read counts can impede the recovery of the arrangements of cells. On the other hand, given the cellular ordering along the trajectory, we bucket the cells by their pseudotime and compute their mean expression for highly expressed and variable genes. Again, for both the full data and the subsample data. With Pearson correlation, we quantify the agreement of each gene's expression pattern. we observe that the estimation of gene expression pattern can benefit from a large batch of samples. Finally, we carry both steps of first inferring the ordering of cells and then computing the gene expression pattern over the inferred order. While each of these sub-steps might individually benefit from either a deeper or broader sampling, our findings indicate that the complete task of accurately recovering gene expression patterns benefits most from an intermediate budget allocation. Okay, so just to summarize and also some outer perspective, we presented here our study of the depth-breadth allocation trade-off in preserving the cellular trajectory structure of a given dataset. We derived models for the reconstruction error when subsampling only reads only cells or both, and for the optimal number of cells to assay. In the manuscript, you can see more examples how these generalize across diverse datasets. We saw that when the sequencing budget is small, its efficient use is crucial. So our results can help in constrained experiments like high-throughput screenings with many conditions. And like our study, there are other studies optimizing experiments towards alternative objectives. So for instance, there is work by Zhang et al. That studies the experimental design towards capturing the statistics of specific genes of interest. When these are lowly expressed genes, capturing many cells at low depth is preferred, which may clash with the optimal setup for trajectory reconstruction. So therefore, as future work, our study can be broadened to address multifaceted objectives and to be posed as a satisfaction problem instead. Okay, so with that, I would like to end and thank my collaborator, Edwin Mehmet, and my PI, Mor Nitzan, and the lab, as well as my funders. Thanks.
Thank you, Noa. So questions?
Oh, maybe I should keep it.
Hi, a very nice presentation. I was curious about other tasks that, based on the estimates that you get, what other tasks can be done? And for example, like cell type discovery, do you think that the estimates that you will get will generalize to other types of tasks? I'm sorry if I missed it because there was a lot of cheering in the background.
Yeah, so there are other works that focus on these kind of studies of recovering cell types specifically. And I think these two things do relate because it does capture like the general structure, but it's not the same, it's not the exact same rules that you would suggest. It would be something similar though.
I see. Can I ask another question? And what about other platforms? Like you did, this is for like RNA-seq, but what about like single cell attack-seq, or like, how much would it actually go to the multi-ohm platform and other modalities? Do you have thoughts about that?
Yeah, so that's a good question. I'm not too familiar with how much like trajectory inference is done with single cell attack-seq, I must be honest. So I'm not sure like how popular that is to actually like order cells by time using that kind of data.
Yeah, people also do it. So
okay, yeah. So yeah, I'm guessing you can also follow the same type of analysis, and it would be interesting to see if it still, preserves like the same type of rules. I didn't try it though.
I would also have a question. So now, in reality, now when I'm in a situation that I want to, make an experiment and I have a budget restriction, how do I use this model? So do I have to say, okay, I think I need, I don't know, I want 10,000 cells, or like Like what is like a realistic application of this? Have you thought about this?
Yeah, so I mentioned it like in the like one of the bullets in the discussion. I think in practice, like because we do need a data set right now in order to suggest like how to perform an experiment, I think it's mostly relevant for like high throughput screenings. So you would do like a one big experiment maybe of like sequencing deeply the cells, and then you can suggest for like smaller experiments how you would assay them in order to still capture the trajectory of cells. So for example, like in high throughput screenings where you might have like many knockouts of many genes, like many perturbations, and you'll have like a pretty small budget for each sample for each condition. I think that might be like a good case of doing it, or when you want to do like some diagnosis over many samples. I think that's also another application where you might want to use this.